Well, thank you. That's a really great introduction. Firstly, can everyone hear me? Is the microphone, it's all good? Nice. So, yeah, it is just a random collection of words. What does it mean? So this is literally my first slide that says, what are you talking about? So DNA. Most of us are familiar with DNA as a biological molecule for storing genetic information. I'm talking about the same DNA that's in your cells that contains your genetic code. So we've got DNA. And then we've got origami. Most of us are familiar with paper origami. You might fold up a piece of paper, in this case, into this little robot. You can follow the instructions here. It's pretty fun. So we've got DNA, we've got origami. I'm talking about those things that you're already familiar with. And then we've got nano. So compared to the talk you saw earlier today, we're talking about really small stuff, not really big things. So what is a nanometer? A nanometer is 10 to the minus 9 meters. So if you think of a human hair, that's about 100 microns. A micron is 10 to the power of minus 6. So if you think of a nanometer, it's very much smaller than a human hair. So that's the size regime we're talking about. So those are the three words, and they're exactly what you might have thought they meant. And hopefully by the end of this talk, you'll see how they might come together into a meaningful title. So don't worry, we're not in a land where you're totally not understanding the use of these words. So I like to start my talks by saying that science is not about one person. I'm here, I'm talking about this stuff, it's really cool, I really love it, but nothing in science happens without teams. And so I like to introduce my teams first. Lots of people put their team at the end, but none of this would happen without my team, so I'm really happy to show them off to you first. So this is my group here at Sydney Uni. We're a pretty small group, but we're growing. And a lot of this work is done by these groups of student teams that are doing a program called Biomod that I'm going to mention right at the end. And that's a way that undergrad students can get involved in this kind of self-assembling nanotechnology and do their own projects. So this is last year's Biomod team, so you'll hear some work from Charlotte and Eric and the rest of the team. And this is the year before. And then we have some really cool pictures coming up from somebody who works at UNSW, the other main university in Sydney, John Berenget, who in true Australian form, the shortening of his name is not John, it's Gut. Took me a while to understand where that came from. I think he likes to eat a lot. And then, of course, a lot of this work, as Chris mentioned, started when I was a postdoc in Harvard. And we really like self-assembly. So in America, you might know that Halloween is a really big deal. You have these group retreats where you go away and you have to come up with the best Halloween costume. And so liking self-assembly, we went as the group Tetris set. And you might see some Tetris-like themes coming up in this talk. So those are the kind of, and so this is me here, and this is the whole SHE group in Harvard. So this is a sort of a team expertise going on here. So I'm going to start with why we would want to make a nanorobot. So I come from this, I read a lot of science fiction when I was younger, and I wanted to make tiny little machines that do cool stuff. Unfortunately, that doesn't get you many research grants, and it's really hard to explain to your head of school. So we sort of thought, what is the purpose of some of these structures? And, like you might build a spaceship to go to outer space, we like to think of saying we're going to build these tiny little machines that are going to inner space. And what is the most interesting inner space? It's inside your body. So if you think this is a picture of a blood vessel, this is like a red blood cell here. How big are these things? So a red blood cell is 6 microns, so 6 by 10 to the minus 6. So we're getting in the regime of nanometers. 6 microns is 6,000 nanometers. So we could make a nanorobot that does something to your cells. And in particular, we're interested in this. So this is what we'd think of when we talk to cardiovascular surgeons or people with heart disease. This might be an early stage sort of beginning of heart disease. And we can't detect this now. And we ran around asking a bunch of surgeons, what would you do if you could imagine being inside the body, if you could shrink yourself down, make yourself really small, and go into the body, would it actually change the information you got and change how you treat patients? And that was a really exciting thing to do, to go and talk to people who have real-world problems and say, what do you want something that could be like a nanorobot to do? And we've spoken in particular to the surgeon, Professor Paul Bannon, who we work with now, and he said, basically, if he could miniaturize himself in the body, he could detect these small changes that signal early heart disease, and that would totally change how we treated the disease. So way less surgery, and way less poor outcomes. And so that's really exciting to somebody who just likes making stuff because it's cool, to say we can actually make something that might be useful to a huge amount of the population. So I think about 30% of people will suffer from some complications from heart disease at some point. So that's the why. And then this is the dream. This is now the future you blood vessel. These are future you red blood cells. And these are future you DNA nanorobots that are going to go into your body, go around, find things that are bad, and fix them so you don't need to have surgery. So it sounds like a pretty far-fetched dream. But hopefully over the course of this lecture, I can try and convince you that it's not as far away as you might think. So there's going to be a lot of interesting representations of things. So when we think about things on the nanoscale, you often have some really glaring things that are wrong with these diagrams. And so one here is like, I told you this is a red blood cell. This is 6 microns. This here is the double helix of DNA. So if you know much about DNA, you know that this is actually 2 nanometers. So that the artist who made this was obviously not really thinking about keeping things to scale. So I will point out to you that when there's some glaringly, obviously wrong things, but you might notice them as well. So you can keep track of those to the end. The other glaring thing that comes up in misrepresentations of DNA is that DNA is a right-handed helix. Even on the covers of really prestigious journals, it's often represented as a left-handed helix. I think I've checked all my slides. I don't think I have any left-handed DNA, but you can also keep track of that for me as well. So that's kind of an artist's impression of a nanorobot. It's a bit wrong. There's some things about it that are interesting. It looks like a nanorobot that I might imagine. But that asks us to ask a bigger question. What might a nanorobot look like? It doesn't have to necessarily look like a macro-scale robot. And this was a really fun project with John Berengut Gut. And we were thinking, well, we could look at macro-scale robots. We could look at robots from science fiction that people draw. We could look at the kind of robots that make cars that don't really look like a robot that has arms and legs that walk around. But we could also take inspiration from the things that already exist. So those are the things in biology. And this is where, as a physicist, it's great to come and look at all the weird stuff that happens in biology that you never might imagine. And sometimes I tell my physicist friends about this stuff, and they actually don't believe me. So we already have kind of nanorobots in us, in the sense that we have immune cells. So immune cells can travel around your body, they can interact with the environment, and they can do things like, in this case, capture bacteria. We also have things like, you might have heard of this, it's called a bacteriophage, and this is a complex kind of robot that carries genetic information, and it's going to attack bacteria. So it's going to find the bacteria, it's going to attach to them, and it's got a little like syringe part here, and it's going to inject its own DNA into the bacteria. So these are the kind of things that inspire us. We see, well, biology can do this. Surely we're smarter than biology. Maybe that's a little bit of hubris there, but maybe we can make something that kind of looks like these things. And so these are the sketches that Gut, this is him here, that he came up with initially. And he used to be an industrial designer, which is really fun to work with somebody who just, I was like, well, we want to make nanorobots, but we don't want to be constrained about what they might look like. Just draw me a bunch of pictures. And he's like, the next day, he's like, oh, I was just sitting in front of the TV and I sketched out 50 nanorobots. This is only half of them. And they might be these weird little octopus things, something like this that looks really simple. Where's my favorite one? I really like this one because it's kind of like that bacteriophage. This is like the squid. So there's all sorts of things, and there's things that might look more like just a real like macro-scale robot but shrunken down. And so it's really nice in this field to sort of let go of what your intuition tells you about what a nanorobot is and let the function of it guide how it should look. But all of these things could be nanorobots, and we could make different varieties of all of these. But we settled down on some things. We had to decide, we're making a pitch. We're saying to some people we want to make nanorobots. We needed a picture of them. What should they look like? And the idea is that they should have some basic things. So a lot of those robots you might have seen, they have legs. So there must be some way to like interact and move around the environment. In this case, we want to make them out of DNA. But you might see here, our DNA is also out of scale. So we've gone for that. To communicate the idea of molecular robots, you need some molecules there. But it's kind of hard to represent them at the right scale. And then it needs to sort of interact with things. So here we have you could have a clamp or a claw or some sort of radar. Maybe it needs a robo spatula to fry its eggs. So there's lots of different functionalities. So you can think of this key concepts that the robot's going to have no matter what it looks like. So then gut being really awesome at 3D CAD rendering, turned those sketches into these amazing pictures that we did a lot of brainstorming about what kind of colors the nanorobot should be. If you think this is on the nanoscale, so color isn't really a concept that makes sense at that scale, but if you're going to show this to, the vice chancellor or people like that, you need to give it colors. So we gave it some nice, calm, medically looking colors to be like, this is a safe nanorobot. It's not going to attack you. So we say, well, this is still science fiction. It's still out of scale. If you say, we're saying we're trying to make something that's 600 nanometers, but here's our DNA, and that's definitely not 2 nanometers. But it's starting to get towards something more realistic. And then we sort of break it down into those components. So we say it needs a body. It needs some sort of scaffolding to hold all the stuff together. That's the core, sorry. It needs to be able to move, so it's got legs, and they need to walk in some way. It needs to sense its environment and interact with it. And it needs to react based on what it senses. So these are kind of the concepts that we need our nanorobot to be able to achieve. And then something that I'm only going to talk about briefly at the end is that it needs to interact. So it needs to interact either with other nanorobots or it needs to interact with us. So for example, if there's something wrong in the body that it's detected, it needs to tell us about it. How is that going to happen? So that's the big picture. These are just pictures, though. How do we actually make them? So this is where we get back to the DNA and the origami. So we say we're going from science fiction, now we're in the world of anatomy. So my talk is going to have 3 major components, and I'm going to only talk about 3 of those 5 functions today. Hopefully we'll get through all 3. So the first is the core. How do we make the core? The second is how are they going to move around? And the third is how are they going to react in complex ways to the environment that they're observing? So those are the 3 parts of the talk. But to understand all of the talk, we're going to have to understand one more key concept, and that's the idea of self-assembling. So I love Lego. I feel like I'm really happy every day because I get to play molecular Lego in my lab. And one of the cool parts about Lego is that it's modular components. But if you imagine you take all these pieces and you want to put them together into a nanorobot, somebody has to do it. Somebody has to pick up all those pieces and put them together. And that's kind of called top-down fabrication, where there is a larger, more complex machine or person that's taking simple components and putting them together. The idea of self-assembly, which is what happens in biology, is say you could design these Lego pieces to be smart in some way. So you're designing them so that they know what they have to be next to in the final design. And I'll talk about how we do that later. But then you could take all your Lego pieces, you could program them in the right way, you put them in a bag, you kind of shake the bag up so you give it some energy, you give it some energy to rearrange, and then you open your bag and your fully formed nanorobot is right there. You don't actually have to go in and move those pieces around. So that's the main theme about how we're making these things. We're being guided by that idea of self-assembly, so we don't need big fancy equipment to make these things. We need big fancy equipment to see that we actually made the thing that we said we were going to make. But we had a photo shoot recently where they for the university marketing, they were like, we want you to stand in front of something that looks cool and expensive. We're like, we don't have those things in our lab. Because actually we're designing the molecules to do all the hard work for us. So step 1 is the core. We need a body for our nanorobot. And I've got a cute movie here. So I mentioned Charlotte from Biomod last year at the beginning. She made, as the Biomod team, they do a lot of stuff with communications. So we set Charlotte the task of saying, how do you explain this to somebody who has no scientific background? And this is what she came up with.
Hi, I'm Charlotte from the UCID Biomod team. And in this series of videos, we're going to be explaining the science behind our project. First, let's talk DNA origami. So everybody knows that DNA contains genes, the biological instructions that make you, you. And most people also have an idea of what DNA looks like, something like this. You've probably heard that shape referred to as the double helix. Many people think the double helix structure of DNA is a single rigid unit, sort of like a ladder or a piece of spiral pasta. But in reality, DNA is more like two ropes or strands twisted together, and these strands can be separated. A single strand of DNA without a partner is, unsurprisingly, called single-stranded DNA. So if it's possible for DNA to unpeel into single strands, what's keeping those strands together in the double helix structure we all know? To answer this question, we need to look a little closer at those strands. Each strand of DNA is made up of a series of individual units strung together. Those individual units are called bases, and there are 4 different types: adenine, thymine, guanine, and cytosine. These bases don't just stack together vertically along the length of each DNA strand, but can also pair up with other bases on a nearby strand, and this pairing is the key to the double helix shape. Think of the bases as 4 differently shaped puzzle pieces. The pieces can connect by slotting together, but only if their shapes match. Similarly, bases can only pair if their shapes, that is, their chemical structures, are compatible. For that reason, the base A always pairs with T, and G always pairs with C. They are complementary bases. If you want to make a double helix out of a single strand of DNA, it's really very simple. All you need is a second strand of DNA whose sequence of bases is complementary. In every place where the first strand has an A, its partner must have a T. In every place where the first strand has a C, its partner needs a G, and vice versa. When you bring those 2 strands together, the complementary bases pair up and bond, and you get a double helix. That's the way nature uses bases to make double helix DNA. But in the lab, you can actually do much more. Let's imagine you have a long single strand of DNA. Now, you create short fragments of DNA that are complementary to small portions of your strand. These short fragments are called staples. When a staple bonds to its complementary region, the strand is forced to fold around it. If you repeat that process in many places along your strand of DNA, you start to generate complicated folded shapes. When DNA is folded like this, it's called DNA origami. With careful design, you can actually manipulate that single strand of DNA to fold into pretty much any shape you want. Amazingly, that includes shapes that have medical applications: boxes that deliver drugs directly to cancer cells, nanorobots that navigate to a certain part of the body when injected, and our creation, a bacterial killer that can replace antibiotics. This is the DNA alien.
So you'll hear more from
Stay tuned for our next video to find out more about what we're creating out of DNA. And don't forget to like DNA Aliens on Facebook and follow us on Twitter.
You can still totally do that, even though it's last year's team. So that's Charlotte's great video explaining DNA self-assembly.
Hi, I'm
There we get it again. And you're going to hear more about their project right at the end and see their final video of what their actual device was. So that idea of complementary sequences, from a physics perspective, we don't need to think of DNA anymore as something that just contains genetic information. We can think of it instead as an information-containing material. So instead of your genes encoding how you grow, we can encode any information we want in DNA. And in particular, we can encode structural information. So what we're doing is we're taking DNA out of its biological context, we're kind of stealing it, if you like, and we're using it for something totally different. And that is to guide the sort of self-assembly of our sort of smart Lego pieces. So if you say we have Lego pieces, they're DNA blocks, and the DNA sequence of them is that information that tells them how to assemble. We can mix them all up together and have them form on their own. So this is a repeat of what Charlotte was saying, is that if you have 2 complementary sequences, so if there's an A here, there's a T there, and you mix them up in solution, they stick to each other. That's really all you need to know about this molecule. And so you'll see some increasingly abstracted versions. I guess physicists, we like to draw everything as a sphere or a straight line. You can see a lot of DNA represented in straight lines here. So in this case, so these are some DNA helices, and this is this guy, Ned Seaman, who kind of came up with this field. He was trying to make DNA crystals back in the 80s. But you're not going to see a lot of DNA double helices, actually, because it's kind of annoying to draw, and we can, to most extents and purposes, just ignore it. And so we draw our DNA as straight lines, and sometimes we draw the sequences, but usually not. Like these A's and C's, they don't really mean anything to a human. It's hard to read, so we take them away too. And we draw our DNA just as these straight lines, and we use color to indicate complementary sequences. So say you have these 4 pieces of DNA, and pink binds to pink, green to green. If you mix these up into solution, you can imagine they'll form this, what we call a 4-way junction. So this is a 4-arm sort of lattice junction of DNA. And then we have these pieces here, which we call sticky ends, and they're unpaired DNA. And so we have purple here and purple there, red here and red there. So these junctions are going to form larger arrays. So you can imagine these are like tiles on your bathroom floor, and you're bringing a large array or kind of a 2D crystal of DNA. And you can do that in 3 dimensions as well. So that's the idea. We program the sequence, and we make the structure that we want. But how do we go backwards? And so the technique I'm going to be talking about most today is this idea of DNA origami. So in that sort of example, we're taking small pieces of DNA and putting them together to make something larger. And that works pretty well, but what actually works a lot better experimentally is this idea of origami. So you might be familiar with paper origami. You start with your 2D sheet of paper, and you fold it up into a 3-dimensional object. In this case, we're taking a 1-dimensional object, this long piece of DNA, which we call the scaffold, and we're folding it up into a complex 2-dimensional object. But we can also make 3 dimensions. So this is how DNA origami works. Say you start here. You'd like to make a square out of DNA, a nanoscale square. You take your scaffold and you raster it across the outline of the shape you'd like to make. And then you design these short synthetic, what we call staple sequences, and they're going to cross-link parts of the scaffold to hold it together. So the whole thing becomes a double-stranded piece of DNA. And so this is an artist's impression of how this thing might happen in solution. We have the scaffold coming in, and these are the staples. Everything's moving around in solution because it has Brownian motion. And so all these molecules are moving, they're coming in. This is showing it as a very deterministic process. Probably it doesn't happen as nicely as this in solution. But all these single-stranded pieces of DNA are going to come and cross-link the scaffold into the structure that we wanted. So that's the idea. Up until now, though, you've only seen pictures. Do you really believe it's true? I'm very much an image person, so if I haven't seen a picture of something, I have a real hard time imagining that it's really true. Is this all just a complicated analogy? So that's the next question. We've made these things. They're in solution. You can't see them. How are we going to see molecules? And I'm going to talk quickly about 2 techniques that are going to come up through this talk. So the first one is atomic force microscopy. And this is really simple in concept, very hard to achieve in practice. The idea is you have an atomically sharp tip, and you drag it across the surface. You're bouncing a laser beam off the back of it, and you're keeping track of where that laser beam is. And that lets you trace out the topology of the surface. So you might see this edge here. And this is what an AFM actually looks like. I did my PhD in one of these instruments. Very difficult to use, but really nice idea. This is where the laser comes down. Here's the sample, and here's that camera that tells you where the laser beam bounces off. And you can get pictures like this. So this is from a paper that came out in 2014. So this is a model of DNA. And this is a picture of a DNA on the surface. So this is dragging the tip across the surface, and you see this kind of what's really quite a squishy molecule. And you can start to see things like the helicity of that DNA. So AFM, atomic force microscopy, is a way of seeing 2-dimensional DNA structures on surfaces. And the other main technique we use is called transmission electron microscopy. So you might be familiar with a light microscope. Most people have them in their schools. You have a light source. You have a bunch of lenses. It's going to transmit light through the sample. And then you're going to either see it or image it on a camera. And an electron microscope is pretty much the same. You have a source of electrons. You have a bunch of lenses. You have a stage where your sample goes through. And then you detect the electrons on the other end and see how the image changes because of the sample. So why would we want to use an electron microscope over a light microscope? And that comes down to resolution. So you might be familiar with the idea that the resolution that you can see in a microscope depends on the wavelength of light. And that's because light diffracts. So as light diffracts, your image gets blurrier, and that blurriness depends on the wavelength. So if we say for visible light, our wavelength is about 600 nanometers, we're really limited to a resolution of 300 nanometers. That's way bigger than what we're trying to make here. We're trying to see something that's 2 nanometers. But what is the wavelength of an electron? Well, actually, the wavelength of an electron, so you can imagine an electron can have particle and wave-like properties. If we consider it as a wave, we can give it a wavelength. And the wavelength depends on the energy of the electron. And so if we make these electrons pretty high energy, so we accelerate them, so they're traveling pretty fast as well, so we have to put in a relativistic correction to the wavelengths, then we get wavelengths in the practical microscope more like 3 picometers. So that's 0.3 nanometers. So we can start to see something that's nanoscale with an electron microscope, where we couldn't with a light microscope. And this is not the exact one that we use, but it's exactly the same model. So this is what an electron microscope looks like. So you have this big tower. This is the electron gun. So electrons are coming down here. We put our sample in there. It's all under vacuum, so we have to put it in through a little tiny airlock. And we focus it with magnets. So instead of having optical glass to focus your light, you have electromagnets to focus your electron beam. But otherwise, it's pretty similar to like a desktop microscope in concept. So those are our 2 techniques. That's going to let us see what do our DNA origami look like. So here's the big reveal. So you put all your things in the tube together, and you do some atomic force microscopy. And these are DNA origami. So if you think here, these scale bars are about, this one would be 100 nanometers. So these objects are 100 nanometers by 90 nanometers. And you can see they kind of look woven. They look like fabric. So we're actually weaving the DNA together. And this is the cool thing is you don't just have to make your rectangle. You can make any shape you like. So you could make a triangle. You just have to change your raster pattern. And this paper came out in Nature, so it's a very prestigious science journal, and they're very serious people. And they didn't like calling this the smiley face, so this had to be renamed as the 3-hole disc. It's much more serious. So we like now in the field to give us a kind of tongue-in-cheek, very boring names to very cute things. So you might see some of that later. And so the neat thing is because this is a self-assembly, we don't just get 1 copy. We don't have to make 1 copy at a time. In solution, you would get 10 to the 13 copies of these. We're just imaging here a few of them, so you have a whole bunch of these in solution. And so if you think of it, all the hard work is in the design, the self-assembly does itself, and you get many copies all at once. It's very convenient if you don't like doing many experiments, which is kind of what got me into it as a PhD student. So what is that design process? If I'm telling you it's all about design, how do we do it? Do we just sit there drawing pictures? Well, kind of, yes. So you'd sit there and you'd say, I want to make a box. And you draw a box maybe like this if you're a good drawer. My drawings aren't like this at all. And you'd say, how big do I want my box? And then you think about how big is a piece of DNA. And then somebody in the field has written a really nice software tool, which is kind of like a CAD program, so a computer-aided design program for making DNA origami. And you can make kind of a box shape. So here, every cylinder represents a DNA helix. We kind of just sort of show them as solid cylinders. And then we look at the cross-section. So this is a cross-section through those helices. So you can choose, like, this is based on a honeycomb lattice. You could do a square lattice, lots of different packing lattices. And then this is looking at the whole thing unfolded. So then you could say, well, this is how my scaffold needs to go, and these are where my staples are. But you can see we don't have any of those A, G's, C's, and T's in there. We don't even think about those. We just click this button. So actually, I've cut it off here. There's a button down here that says Add Sequences. And then the program spits out a bunch of DNA. And then we look at this and we say, well, yeah, might be right, sure. Who knows? I mean, nobody could really look at this and analyze it. So then we have to do the experiment to see, does it actually make what we thought it should make? And how do we get our DNA? Like everything else, we buy it on the internet. So you go to this website, IDT DNA Technologies, and you cut and paste your Excel file into their website, and you give them some money. So a whole DNA origami might cost about 1,000 Australian dollars. And then you say, and they send you these nice DNAs in 96-well plates. So they're all like, you might order 300 staple sequences. They're all different, and they painstakingly put them in separate containers. And you take the lid off, and you mix them all in together. And then you just heat it up and cool it down slowly with the scaffold there. And you can do that in like a thermocycler, like a PCR machine that has a very fine control of the temperature ramp. Or you can just heat it up and put it in a styrofoam box. You don't actually need anything sophisticated to assemble these. And these are the types of 3D structures that people have made. So I thought I'd take you on a tour of my favorite cool pictures of DNA origami. So this is the first 3D DNA origami. So this is this idea that once you have that 2D rectangle, you can treat it like a piece of paper, and you can fold it up into the third dimension. So this is a piece of paper, and we're going to bend it twice, and then we get this block of DNA that was on the previous slide. So you can put your staple cross links to hold that 3D structure together. So you get a block. Block's kind of cool. You can make blocks that have parts that go in different directions, so you can get a cross thing. That's kind of fun. What else could we do? So we can actually put curvature into the structures. So by controlling, and this is where we stop forgetting that DNA is a double helix, and we remember that it's a helical molecule. And if you selectively under or overwind regions of it, that it's going to bend, it's going to be strained, and it's going to bend in response to that. So you can make things that this is starting to look like something cool to nanocog. So these scale bars here are 20 nanometers. So that could be part of a device. And this is probably the first nanorobot that anyone made. So people in this field, we like making stuff that's cool. And I think one of the reasons for that is you could accidentally make something, you could convince yourself that you accidentally made this, but really no one could accidentally make a nanorobot. So this is the first nanorobot, and it's kind of waving to you. This is a TM picture of it. You can see its legs and its arms, so it's saying hi. It doesn't really do much yet, but that was the very first nanorobot. And so yeah, this scale bar here is 20 nanometers, so they're really small. And then you can make DNA origami in lots of different ways. So this idea of, instead of weaving fabric, you could weave a basket. So this is kind of basket weaving, where you have the DNAs going around and around here. And this is a DNA vase. You can also make wireframe structures the same way you would animate things in geometries and software with wireframes. You can make, we also really like spirals, DNA people. We're really into making spirals, so they made a spiral. This is the next nanorobot. He looks kind of a bit less structurally sound than the previous one. He's kind of more like, help. He has a nano Coke bottle to take with him, and he also has a nano bunny rabbit. So the bunny rabbit is a really famous thing to render in computer graphics. So they rendered here this nano bunny rabbit. And then you kind of run out of things to make. So in this paper where they called them DNA bricks, where they were taking the Lego analogy further, they made 100 shapes. And after you do the alphabet and the letter and the numerals and other symbols, you run out of, and they really wanted to do 100, so they were like, well, we'll have a space shuttle. So this is a nano space shuttle. So it's not going to outer space, but if you think of this here is 30 nanometers there. And it's going to go to outer space or inner space, and it's going to find some nano aliens, and then maybe they fall in love. So you can make all these shapes out of DNA. And that kind of brings me to the question of, like, what should we make? It's really hard to decide. And this is another poor representation of DNA. So this is a DNA robot made of DNA, but somehow it's tearing a giant piece of DNA apart. And this is, I say this is even more 3 dimensions, because it's not quite 4-dimensional yet, but this is a robot that has different states. So it's starting to be the first thing that could actually do something, but nobody's actually seen it operate yet. So I'm not sure we could totally say it's 4-dimensional. But this is a robot that can flap its arms. So you can see here it exists in many states. This is arms up, this is arms down, and it does that in response to salt concentration in solution. So this is, if somebody showed me a movie of this, then I would say it's 4-dimensional, but there's no movie of this guy yet. So what can you do with these? In the current state of field, can we put them in the body? Can they do stuff? Can that robot flap itself around your body? No. That's still pretty far off. But we can do some other useful things. So you can think of these DNA origami objects as ways to make tools. And as somebody who comes from physics to the weird world of biology, it's really annoying. Stuff moves around all the time. It gets in the way, it sticks to everything. You can't just go in there and hold it down. And so what I got excited about this field is saying we can make nanoscale tools like tweezers and springs and things to, like calipers, to go into biological systems and make stuff calm down so you can figure out what on earth is going on. So this is an example of some work we did where we made, this is a DNA origami spring. And we were doing exactly what you would do with a macroscale spring. We were using it to measure forces, the same way you would weigh your luggage with a spring balance, or maybe weigh some groceries. In this case, we're measuring the force exerted by a molecular motor myosin. So this is one of the motors that transmits things around your body. And we could measure piconewton forces over nanoscale movement with our DNA origami spring. So they're good for measuring really complex small stuff in biology. They're also, like, people are really inspired by this to make smart drugs. So instead of giving chemotherapy to your whole body, you could have a box that contains some therapeutic agents. It could be closed or it could be open, and that it only delivers the drugs to where it needs to go. So there's a whole part of this field towards smart drug delivery that I'm not really going to talk about much. And then the last kind of category of what we would do them with is to control synthetic reactions on the nanoscale. So instead of having your flask or your test tube, that's a really big vessel, we want to make nano-reaction vessels and tether, say, chemicals or enzymes in them to have really small reactions going on. And so kind of like what happens in your mitochondria or cells, you can have really small reactions. So those are the things we currently do with them now. So that was how we might make the body of a nanorobot. The next part I'm going to talk about is how do they move around? So I said we had something that was almost four-dimensional. But right now, we're kind of just making stuff that sits there. We want it to move around. And so I'm going to bring in some ideas about DNA machines. Where are we going? OK. So this idea of DNA machines. How can we make a DNA object so we have that complementarity? How do we make things change? And this is a really simple idea, and it's based on the more base pairs you make, the lower the energy of the system. So when you make base pairs, you're releasing energy. So thermodynamics tells us if there are more base pairs that can be made, the system will go in that direction. So say you start off, in this case, pink is binding to pink, and you have a certain number of base pairs, and you have what we call this green region, the sticky end, or sometimes we call it the toehold, because it's where you can kind of get your toe in to change the system. And that's unpaired, so that's ready to make some base pairs, release some energy. And you add this strand that is complementary to both the green region and the pink one. The green region is going to bind here, and then it's actually going to kick off the small pink strand. So energy tells you that this is more stable than this. So when you add this, the system will go towards this state. So that's a really simple concept of how you can have change in these systems by designing it to go down an energy gradient. And you can do other things, so you don't just have to add one strand, kick off another. You can start with, say, this is from 2000, the first DNA device, which was kind of like a really simple tweezer. So you have these two DNA helices here. You have a strand that can come in and stick them together, close the tweezers. Then you have another strand, so there's another sticky end here. You can strip off that strand, and then you can go back to the open state. So you can cycle things through different states. That's kind of cool. But that's not really a machine, is it? And so this is some work I did in my PhD about using these concepts to make a motor, make something that moves around. So here we have some DNA, what we call stators or anchorages. So these are sites that our DNA cargo here in pink can bind to. And when it binds here, it makes double-stranded DNA, and an enzyme recognizes that. The enzyme comes along and cuts the DNA here. Then we have a small piece of DNA. And at room temperature, the energy of this DNA is low enough that room temperature is enough to melt it. And so actually, this is just going to float away. And then we've revealed a toehold here. The toehold can bind to the next stator. And then this process of exchange, which we call branch migration, is going to happen. And eventually, it'll move over to the next site. But then we've made a new cutting site for the enzyme. So this is, we call it a burnt bridges motor, because it moves forward because it's cutting parts of the track off behind it. So it's destroying its bridge as it goes along. And you could take this and put it on DNA origami. So this, maybe you can't see this very well. This is supposed to represent a DNA origami tile. So it's 70 nanometers long, this little track. We put the motor at the start, we add the enzyme, and we kind of watch it go along. So this is an AFM picture of it. So we put these registration markers here so we can see the track. Actually, if it's single-stranded DNA, we can't see it very well. The track is down the middle here. And we can see the motor as the bright spot. This is, I guess, getting back to that visual thing. Unless it's moving, it's not really happening. So these are movies using the AFM of these DNA motors. So this is the motor here, and it shuffles between these two sites, and eventually it jumps forward. And this is what's called a chimograph. So we're looking at what happens along this line as a function of time. And so you can see there's these 4 anchorages, and it's just a short part of the track, but eventually it's moving from one edge to the other. And we can actually see the stochastic behavior so that it's not moving in a really determined way. It's kind of uncertain. It's not really not sure which direction it wants to go. But eventually it's burnt the track behind it, has to keep moving forward. So that's how you make a DNA motor out of DNA. And then, of course, you don't want to just make it go on a linear track. We've made some little mazes. So in this case, it's a pretty simple maze. We have 2 bifurcations, and then we have a motor. We load it here, and we put gates here. And we can either unlock the gates ourselves, so we block those states by hybridizing different DNA to them, or we give the motor a key that can go along and open the right gate. So we might program a motor to go left or right at this junction. And we're going to see how this motor walks by putting a fluorophore on the end of the track and a quenching molecule that's going to dim the fluorescence at the end of the track. Oh, sorry, on the motor. So here we start here, and then if we go left, would quench red, and if we go right, would quench blue. So in this case, we've added the instructions to quench right. Oops. And so we see the blue signal go down, whereas if we tell it to go the other way, we can make the right signal go down. So we can make these motors that walk around complex environments. So that's kind of the bones of how our DNA origami motors would start to walk around. And there's some links to how these walk as to what cellular motors are doing in your body. So step 3 is react. And I wasn't sure if this was going to fit in time. I guess I got 5 minutes. I'm going to go through this pretty quickly. And there's quite a lot of equations here, and it's late on the first day. So I'm happy to talk with anyone about this offline who's really interested in circuits. So how are we going to have complex information processing happen for this robot? And I'm going to talk here about some really beautiful work done by Lulu Chen and her co-workers in Caltech. And a lot of these slides come from some of her work. And so this is the idea that we have electronics. And with electronic circuits, we can do amazing things. We can make self-driving cars. You can make your phone or your laptop. Why aren't we doing that with molecular computing? So what is the molecular equivalent of the electronic circuit? So say, in this case, this is a genetic circuit. You can think of parts of biology as acting in the same way as electronic circuits. And if you have molecular circuits, you can use molecular programming to make really sophisticated behaviors. So the simplest circuit that I like to think of is an AND gate. The AND gate is really simple. You have 2 inputs, A and B, and an output. And the output is only 1 if both inputs are 1. And most of us are familiar, we learn this in school pretty often, that you can make an electronic AND gate out of transistors. How would we do this out of molecules? And we use that idea of having sticky ends on our DNA and using toe holds to achieve an AND gate. So this is some work from Lulu's lab where here we have our input strand 1 and 2. And then we've designed this set of DNA molecules that's going to interact with them. And these have a toe hold there, and these have the same toe hold there. So the input strand is going to bind, and it's going to kick off this other strand. It's going to displace it. That strand is then going to go interact with some other strands. And this one, we call this an annihilator. It's going to cancel out the signal. If you think of this as the input, it has no output. And then there's also the output strand. So if you have 2 molecules of this, one will be taken up by the annihilator, the other one will be processed into an output. So this is the output. That's how it's operating as an AND gate. You could imagine if you take out the annihilator, it will operate as an OR gate, because you only need 1 input to produce the output. And then this is some data to show that does actually work. This is, again, using fluorescence quenching, where here we have a fluorophore that's quenched. But if the output is produced, it can bind here to the fluorophore, displace the quencher, and we see a signal going up. And so this is the signal of that fluorescence over time. And depending on which input you have, the signal will only go up if the AND gate is true. So we've made a molecular AND gate. And the next bit is pretty hand-wavy because there's quite complex circuits in here. But you just need to get the gist of it, of say, what kind of information processing can be done with the molecular AND gate? Well, AND is kind of interesting. That's nice. You can say, if I have this receptor and that receptor on my cell, I'm going to release the drug. But surely we want to do something that's way cooler than that. So this is the idea of saying we can start to program sort of neural network algorithms into molecules. So this is a nice neural network example. It's called the winner takes all. And some people like mathematical representations, other people don't. But the key take-home message here is this. If the output bit has value 1, if and only it, the corresponding input is the largest input. So if you give it a set of inputs, it will tell you which one is the largest one. So that's the winner. And so this is a mathematical way of expressing that some people might be more comfortable with. And these kind of neural networks are really useful because they're great for analyzing images. So say you have a piece of handwriting, and you want to know, is this thing more like a T or more like an L? It's not exactly like either of them. This network gives you a way of assessing what is it most similar to. So this is an idea of having a target pattern as a weight. So again, this kind of representation might be comfortable for some people or not otherwise. Also, I'm having some interesting symbol replacements there. Thank you, PowerPoint. But here's a visual representation. So say this is my input letter. Oh. So this is my input letter here. And I want to know, is this more like an L or more like a T? So I have my weights here. So these are my weight functions, and they say, well, this is what the canonical L looks like. And this is what the canonical T looks like. And if I multiply the corrupted pattern, my input, with whatever I've said on my weights, and then I do the weighted sums, and so this is what that would look like for the thing, because this does look a little bit more like an L. And then you say, well, which one of these is highest? So your winner takes all helps you decide, is it more like an L or more like a T? And so you can imagine this is like just an algorithm. You could encode it in software. You could also encode it in AND or in OR gates. And what do those AND and OR gates look like? Well, a little bit more complicated than the last one. So obviously I'm not going to go through this here, but you can look up this paper. It's a really lovely paper, and it's explained really well to say these are the circuit networks that do those calculations to say, is this an L or is this a T? And as you might imagine, you can encode these with transistors, but you can also encode these with molecules, and that's what these people have done. And they didn't just do it for something really simple like an L or a T. They actually did it as a handwriting analysis of numbers. So in this case, they have all these different, and these are kind of class averages of different versions of the same numeral. So this is kind of, this is the average of 1. This is the average of 8. So somebody's drawn these, and you can create weight functions based on those handwriting averages. And then when you have your input molecule, so say you have a particular numeral that's been written, it's been expressed in molecules, and then you can do those multiplications. So you can multiply your example against all the different weights and say, is this most like, and this answer comes out here, it's most like a 7. So this is how you can use molecules to actually analyze complex behavior and do complex information processing. Is this going to be faster than your computer? Never. But can you put your computer inside a body? Probably not. So the idea is that this bridges the gap between modern computing and what's biocompatible computing. So could this be looking at the inside of your blood vessel and saying, in this region, is there enough cholesterol deposited that you might get heart disease? And then compute the answer based on something like a weight function. So that's the idea behind how these things are going to react to complex situations. So I like to think about what happens next. Of course, we want to dream big. We can only make our little nanorobots so complex, so we'd like them to interact. So these are the ants. They're making the bridge. They're doing well, and then the bridge moves away, but they actually have a really good strategy for fixing the problem. And it's not like really obvious that the ants down the bottom know what's going on at the top. But somehow they're able to know when they can come back up. And there's really simple rules that you can program into these systems that people are starting to explore that allow you to do this kind of thing, firstly with like little micro robots. So they have this thing called the Kilobot that you could Google, where they have these robots. They're very simple rules. It's kind of like, if you see another robot on the left, keep walking. And you can use them to make complex shapes by encoding simple rules like that. Same way, we would like to do that. Oh no, this is... But they recover. And they can also do things like find the shortest path to somewhere. Okay, so our idea is that in the future we want to take advantage of those algorithms that do those complex emergent behaviors with our nanorobots so we could have a swarm of them in your body doing complex things. So if you're interested in this, and this has sparked your imagination, how could you find out more about it? How could you get involved? So Biomud is where I want to finish today. So Biomud is a great thing because a lot of research gets done by people who are doing PhDs. And as an undergraduate, maybe you're in your first year of your degree, you finish school, you're like, I want to do physics, I want to do chemistry, I want to do biochemistry, but I don't know if I want to do research. Do I want to do research? Biomud is a great way, along with other sort of programs similar to it, that you can get research experience while you're an undergraduate. So this has teams of students from first year, second year, third year, all different parts of all different faculties. So on Biomud, we had Charlotte, who was an art student, we had engineers and scientists. And you come together and you invent your own research project and you tell the world about it. So this is them, they compete as teams in California. So our Biomud team won the best presentation last year. So you compete against a whole bunch of other kids who like nerdy stuff and like to, in this case, this competition is for building biomolecular stuff. So the only rule of the competition is make something out of a biomolecule and have it do something. And so it's really open-ended, and that's really exciting. And so you come up, it's really fun to look at, and you have to present a website, you have to make a YouTube video explaining what you've done. And so their website, the Biomud website, is a really neat way to look at all the kind of cool things that people have done. So in this case, if you're familiar with paper origami, this is a thing called a murafolding. And they've actually made a molecular version of the murafolding. So it's a way to make very collapsible structures. And this is our team from last year that you heard before. They're the Dean Aliens. And so I'm going to finish with their final, Eric's masterpiece. He'd be very embarrassed if I was showing. He knew I was showing you this. So this is the video they presented in San Francisco to explain their concept.
Scientists, we gather at this time of crisis for a top priority mission to save humanity from a dangerous threat, antimicrobial resistance. This is the enemy, bacteria. We have been fighting them since time immemorial, and for decades they have been growing resistant to our antibiotic weapons, despite all of our advances. In 2016, bacteria killed nearly 700,000 people. Their rampage increases every year. By 2050, they are projected to kill more people than cancer. They must be stopped. Our researchers have been designing a solution. Your mission is to help them build it. Inspired by the shape of ancient alien technological relics, we have designed a new weapon to attack bacteria, the DNA alien. Here are the blueprints. At the heart of our design is a DNA origami pore. This will insert itself into the bacterial membrane, creating a physical hole, disrupting the bacteria and killing it. This DNA pore is destructive and needs to be hidden so only harmful bacteria are targeted. We hide this pore inside a protective barrel made from DNA origami, a DNA scaffold that repeats itself along its circumference with staples that hold it together. The pore is only released when it reaches the bacteria. The release mechanism is via aptamers, special DNA strands that recognize specific bacteria. Once recognition has occurred, the DNA nanopore is unlocked and released from the barrel for destruction. Here's the plan in action. The aptamers will bind to the bacterial surface. This will trigger the top locking strands to release the tether, allowing the pore to move around and find the membrane. When the pore finds its target, hydrophobic moieties on the pore will allow it to insert into the lipid membrane of the bacteria, and molecules will start to flow through. Finally, to keep the hole open, the pore will be held in place by lower locking strands. This will destroy the bacteria. The time approaches to make these blueprints a reality. Scientists, only you can save us from bacteria now. Well, only you and Team DNA Aliens!
I meant to say, not only did they do all those animations themselves, they did all of the science that actually made that thing, and they started testing it with liposomes. Eric wrote the whole soundtrack as well. He plays 22 instruments. He says poorly, but I think he's pretty good. And so that's the kind of thing that we're running here at City Uni, and that there are like teams all over the world that do this kind of thing where you can get involved in not just doing some science, but coming up with your whole rationale. So this was a project I never would have thought of. It was so successful that now we've written some grants on it, and we're actually pursuing it, not necessarily to destroy bacteria, but to interact with any other lipid-bound objects, so like cells. So I'm going to finish there. That was such a wonderful video. And do you have any questions? Do you have any ideas of things that we should make on the nanoscale? Thank you.